Hello, everyone, and welcome to today's webinar titled Accelerate and Simplify Spatial Proteomics for Translational Research with the EVOS S1000 Spatial Imaging System. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com/spatial-biology. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Adyary Fallarero, Ph.D., Senior Product Manager, Thermo Fisher Scientific. Adyary, you may now begin your presentation.
Good morning or good afternoon, wherever you are in the world. I'm pretty excited to be here with you today presenting in this webinar. My name is Adyary Fallarero and I'm coming from Thermo Fisher Scientific in Finland. This is the 6th edition of our Fluorescence Academy, and this webinar is being presented in partnership with LabRoots. Our goal today is introduce you to some of the newest innovations that Thermo Fisher has developed to accelerate and simplify spatial proteomics research. And we are truly excited because spatial proteomics is an incredibly thrilling new area of life sciences. And whether you are new to the space or whether you are already in the space and are a very sort of knowledgeable spatial researcher, we hope that this webinar can be of use for you. So thank you in advance for joining us during the next 1 hour. So let's get started with the topic that we are going to be covering today. So this is a webinar in the context of our Fluorescence Academy. So therefore, we are going to be talking a lot about fluorescent imaging, but we're going to be doing so in the very specific context of the spatial biology revolution. So we are going to talk about what spatial biology is and why it has become such an important new field in life sciences. Then we are going to home into spatial proteomics, so specifically on trying to understand how to perform spatial protein mapping in tissue and why multiplex fluorescent imaging is such an important tool that we can deploy. And then I'm going to introduce you to the new tools that Thermo Fisher Scientific has developed in this space. I'm going to start with our new instrument, the EVOS S1000 Spatial Imaging System, and I'm going to kind of show you why we believe that this instrument can truly help you accelerate and simplify research on the space. And then I'm also going to present some of our new labeling solutions for protein tissue mapping that can be used with the EVOS S1000. And I will end up with a case study as a result of one of our collaborative efforts within this program. I probably won't have time to go through every single product and every single new innovation that we have on the space. So I have included here also a QR to our Spatial Biology Research Center. So I will really encourage you to visit the website and gather a lot more information that we have prepared there. So as I said, what we are discussing today is fluorescent microscopy in the context of spatial biology. And for those of you that are new to the space, spatial biology is all about trying to understand the distribution and localization of single cells in the tissue microenvironment. So it's all about trying to understand the diversity of cells and how they are organized within tissue. It's all about trying to understand the different cell types, the different cell functions, how the cells interact, how they form networks or very specific patterns and correlations, and kind of trying to see how that becomes relevant in the context of the disease progressions, in the context of different disease stages, and also in the context of therapeutic vulnerabilities and therapeutic responses. So it's all about trying to understand what the differences are on tissue that can explain the response to certain drugs and really the progression of certain disease in general. The spatial biology term also embodies a number of different technologies, all kind of encompassing a spatial single-cell omics. And today we are specifically going to focus on the proteome, so particularly on the spatial mapping of protein in tissue. This is an area that we call spatial proteomics, and spatial proteomics has become very popular, and it was in 2024 recognized as the method of the year by Nature Method. It was following really multiple, like an evolution of multiple years from 2020 all the way to last year. And we're pretty excited about all the new venues for research that are being opened up. And we are therefore also very excited about the opportunity to contribute to this space with the new tools that we are developing and that I'm going to be discussing in this webinar. So let's now dive into the methods that one can utilize to study spatial proteomics on tissue. And I think it's very obvious that one of the most widely used methods is multiplex fluorescent imaging. From the methods that you can see here on the slide, if we start from the left, I believe that most of you are familiar with conventional microscopy. Those are methods that have existed for a long time. They have been used for live cell imaging as well as for tissue imaging. And I bet that many of you have performed at least once a 4-plex type of study where you have been very easily and in a very fast manner, you have been able to visualize 4 targets in a tissue. The advantage of that approach is that it's very straightforward. We are all very familiar with that, but the limitation is that you cannot really capture the diversity in the tissue by using such a low number of targets. So it's not enough to capture all the biological diversity that exists on the tissue. Conventional microscopy can also be utilized in a slightly more innovative way, and now we are talking about the methods that you see right in the middle, the cyclic labeling and imaging. Those are methods that are kind of performed in an iterative fashion. So in every cycle there is between 2 to 4 targets that are labeled and then subsequently imaged, so that you can perform this in multiple times with the great advantage that you can interrogate a very high number of targets. So the more cycles you perform, the more targets you can interrogate. That is obviously great from the perspective of the cellular diversity that you can capture, but it has some drawbacks. The main one is that it's a low throughput technology, and because we are performing multiple cycles and there are stripping in every cycle, well, such cycles can damage the tissue, and because you're also performing this in an iterative way, then there's always the need for post-acquisition processing steps where you can co-register those images and generate the full composite with the full plex that have been kind of generated. I have to say the cyclic labeling and image has been really instrumental in advancing spatial proteomics in general. A great number of studies that have been more of the discovery nature have been performed using cyclic labeling and imaging. But it's not the only technology that exists. There is a third technology that is basically the focus of our webinar today, which is spectral imaging. And spectral imaging uses a slightly different approach. Rather than focusing on a discrete number of targets, like cyclic labeling and imaging does, so only 4 by cycle, up to 4 by cycle, spectral imaging expands the number of targets that can be captured in a single round. And in general, we can capture much more than 4 fluorophores in a single round. Because we capture so many in a single round, there is no need for stripping steps, and the illumination of the tissue is also much less. Therefore, there is much less likelihood that there would be photobleaching, and there is consequently better tissue preservation. But of course, there is also a drawback, as it is in all of the cases, and it is that because we are imaging simultaneously multiple fluorophores that are very close can, of course, have signal bleed through, and that signal bleed through needs to be taken into consideration, and we are going to be discussing very briefly how that's done just shortly after this. What I want to highlight is that because each method has pros and cons, I think the importance is here is that
the right selection is done depending on the biological question that you have at hand. Spectral imaging it's ideal for a certain type of research that is more hypothesis driven, when you don't need to interrogate a large number of targets, when you know specifically the discrete number of targets that are interested for you. Then I think that spectral imaging is an essential tool that can provide a lot of valuable information for your research. I mentioned in the previous slide that spectral multiplex fluorescence has the great advantage that we can detect multiple fluorophores simultaneously in a single round. But the drawback of such approach is that because fluorophores tend to have this broad emission spectra, that results in spectral overlap and signal that can bleed through different channels. In the example that you see here, if we look at the top panel of fluorescent images, you have the 514 signal associated to ECAD bleeding to both the 488 channel as well as the 555 channel. So it does kind of confound the images, and you cannot really simply discriminate between the targets because ECAD shows up in all of the three channels. So to resolve that problem, there is a way in which we can discriminate, and that is by using spectral mixing. And spectral mixing, in a nutshell, it's a mathematical processing that can be applied to establish the specific contribution that each fluorophore has to every pixel of the image. At the end of the day, every image, it's a combination of spectra coming from all of the fluorophores that are on that finally plexed image. And if we understand how every individual fluorophore behaves, then we can easily resolve this signal and apply what we call an mixing metrics. So in that scenario, when we apply that on mixing metrics, protein targets can be resolved even if they are fluorescent signals overlapped. You can see in the example here that when the image is unmixed in the bottom panel, ECAD then it's exclusively present in the 514 channel, and it doesn't show up then on the neighboring 488 or 555 channel. So even though there is this limitation, it can be easily overcome by using spectral mixing.
Now that we have understood what spectral separation or mixing is all about, then I think it will be easy for you to grasp and understand what the innovation that we are bringing to you is about. So the instrument that we are now launching, and that it's the sort of the core component of our presentation, it's the Invitrogen EVOS S1000 Spatial Imaging System. And this is an instrument that utilizes a spectral technology to be able to perform single round imaging for a very fast tissue protein mapping. And it's able to detect simultaneously 9 targets. We're talking about 8 protein targets and DAPI, so nuclei staining, in one single round of imaging. So it's able to, of course, perform this spectral separation and generate an image that very accurately describes the target localization on the tissue. How is this different from the existing technologies is what I'm going to try to demonstrate to you. And we are going to do that by first understanding what are the current challenges that spectral images that are existing in the market have. The first one is that this spectral separation or mixing that we have been discussing about, it usually is limited to certain vendor-specific or proprietary dyes. And that is, of course, limiting the capabilities that you can use the instrument for, and it also limits the type of targets that you can detect. So it sort of restricts the capabilities of the labeling solutions that you can utilize. And that is the first problem that we really wanted to address. The second issue is that in general spectral separation, it's done with a sort of a black box or cryptic approach, which is not necessarily something useful for scientists. Scientists like to understand what the outcome of the mixing algorithm is. And again, this was another challenge that we wanted to address. The third challenge that we're going to be discussing today is the fact that for the most part, the instruments that are currently existing are very difficult to utilize and are very difficult to implement the on-mixing workflows when you go beyond 6 protein targets. And because the EVOS S1000 can detect up to 9, we wanted to make equally simple the process of detecting 6, 7, or 9 protein targets. So we wanted to emphasize on the simplicity of the workflow regardless of the amount of targets that the user is detecting. And that lead us to the last point, which is like that we really wanted to make it as easy and as intuitive for most of the user. It's particularly critical with a new discipline. If we are talking about a new discipline in life sciences, we have to make it easy to adapt. We have to make it easy for new users to come to the space and very easily find their ways into acquiring high quality or high resolution images. So now let's go through each of these challenges, and I'm going to show you how we have been able to address those challenges and implement better solutions in the EVOS S1000.
The first benefit that I want to highlight is the flexibility of the system to utilize different kinds of dyes, unlike other systems that are in the market. The EVOS S1000 is flexible to use with Alexa Fluor, Alexa Fluor Plus, Allura, as well as Opal, special amplification dyes. And on-mixing has been validated for those fluorophores as well as for spectrally compatible fluorophores. The user just needs to select a fluorophore from a drop-down menu. And once the selection has been made, then you are presented with this protocol setup that you see here on the screen. And the channel configuration is automatically created for that specific plex that's been selected. The emission spectrum is shown and the channel configuration is also shown, so both the primary channels in blue as well as the support channels. So the user doesn't need to make the decisions related to the channel configuration that is needed for a mixing. And I also want to highlight that it's compatible with both primary conjugates as well as amplification solutions, even with a combination of both. So I'm going to show you some examples of that in the following slides.
And why having these reagents flexibility is important, it simply is because it allows for versatility on target detection. In the vast majority of the cases, the expression of certain targets will vary from different tissues and from different disease conditions and between different patient types. So it is absolutely important to have a system that allows to detect a great range of different expression levels for the targets of your interest. So with the EVOS S1000, there is opportunity to use primary antibody conjugates, so primary antibodies that are directly labeled with a fluorophore. And in that scenario, then you can add those antibodies very quickly into the tissue, incubate for a very short period of time, and that labeling will be very effective, but for the most part with medium to high expression targets, as opposed to the other sort of end of the spectra, when you would have potentially targets that are very low expression that would therefore require a labeling system that allows for the signal to be amplified. Here I'm giving you the example of the Allura special amplification. This is in general a pyramid-based amplification workflow that we are going to cover later during the webinar. But what it offers really is the opportunity to use any type of labeling detection methods based upon the expression of the targets that you would be interested in detecting. So providing this type of reagent flexibility allows for scientists to pick whichever is best suited for their tissues and for their target expression levels, and there wouldn't be any restrictions on how they can be used with the EVOS S1000 and the possibility to separate spectra and perform on mixing with this instrument.
A second distinctive aspect that we have tried to address with the EVOS S1000 is the lack of transparency on mixing pipelines in current spectral imagers. In the case of the EVOS S1000, when a protocol is set up, as you saw in the previous slide, if on-mixing is needed, we will encourage users to acquire images from individually stained samples as well as from unstained tissue samples. And with that, the workflow will extract spectral information and generate an on-mixing metrics. And as part of that protocol, it's also possible to generate an on-mixing quality metrics report. That on-mixing quality metrics report contains both qualitative as well as quantitative information on the quality of the mixing even before the area scanning has been initiated. You see an example here of the type of information that we show, but there's also quantitative parameters that the user can simply look at, and by following certain established thresholds that are are shown on the report, it can easily judge in an objective manner whether the quality of the on-mixing is good enough before even the scanning has started. There is also troubleshooting tips that are provided on the report, and overall we feel that by using this report, which gets to be associated with each protocol, we can really increase the confidence on the quality of the on-mixing process for better use of the spectral imager.
The sub-challenge that we wanted to address now here is the fact that for currently existing spectral imagers in the market, any on-mixing workflows that involve more than 6 protein targets is truly very cumbersome. With EVOS S1000, we've tried to address that, and we've tried to simplify and accelerate the way in which that can be done. So first, we go through the entire single color controls workflow, which was described in the previous slide, so that the mixing matrix is generated. And once that protocol has been created and once it contains the mixing matrix, then it can be loaded into the software, into the EVOS S1000 Spatial imaging software, and you can start then scanning your samples of interest. In the video that it's going to start playing on the screen, you can see how the user first can set up any area scan of any size, so it can be as big as 1 square centimeter or bigger, or it can be small. In this case, it can also be placed in any location within the tissue. We are now zooming into a 2 square millimeter area that we have defined. It's a rectangular area. Then the system will start autofocus as soon as you hit capture. And once the first field of view has been autofocused, then that will be maintained throughout the entire area scans. Here we are doing a 9-plex, so we are acquiring 16 channels, the 9 primary channels and the 7 additional channels that are needed for a mixing. But what you're seeing is that every field of view is acquired 16 times, and then the last flash is the fully on-mixed field of view. So by the time the image acquisition has been completed, the outcome that you see that is being generated is a fully on-mixed, fully stitched OMET file. Now you're ready to visualize and explore the tissue, which is what we're doing in the video. So we are zooming into the tissue. We are seeing all the different single cells identified with the different targets that were included. You can then isolate specific channels, for example, in this case, DAPI. Then we can go through every single individual channel that has been included here with the different fluorophores. And what you can also appreciate is that there is no bleed through between the channels. The on-mixing has generated this bleed through that we were referring to at the beginning. So this is the great benefit of spectral imaging. So by the end of the image acquisition, you have a fully ready-to-go image that has been completely on-mixed, completely stitched, and it's ready for downstream analysis in any third-party software that supports standardized OMET files. So it really is the fastest that you can get to a fully ready file for spatial analysis. And I didn't mention timing, but the example that you were looking at, it's a 9-plex done at 20x, so the most common magnification used for tissue imaging. If we were to scan a fully 1 square centimeter of tissue, then the time that it would take to have the fully on-mixed and stitched image, the outcome, would be less than 1 hour, around 45 minutes for the whole image to be generated, means on-mixed, stitched, and saved. So that is how simple and fast the instrument can be.
And last but definitely not least, the EVOS S1000 is an instrument designed, taking into consideration even unexperienced users. We pride ourselves of having biologists that are designing instruments for biologists. And it also builds on the EVOS sophisticated simplicity. EVOS instruments were the first digital microscopes that entered the market more than 15 years ago, and they were built with enough sophistication so that even very inexperienced microscopists could obtain really high quality images. And that's exactly what we want to do also for spatial. Simplify it to the point that even if you're new to the space, you would be able to acquire really high quality spatial proteomics images.
We have covered the main features of the EVOS S1000 and how it can accelerate and simplify spatial proteomics imaging. We are going to focus the second part of the webinar on the labeling solutions. So we have highlighted that the EVOS S1000 can be compatible with any sort of labeling solutions, and it really is not specific to any vendor dyes. But we are going to introduce you to some of the newest innovation that our company has developed in the space, and how these innovations can be utilized in combination with the EVOS S1000. And I want to start with the Allura special amplification reagents. These are reagents that were launched recently and that utilize a pyramid special amplification as the sort of detection workflow in which the targets can be identified and amplified very effectively. The workflow is pretty straightforward, and you can see it here on the slide. It starts with a primary antibody. This is a primary antibody that can be any primary antibody, as long as it is mouse, rabbit, or biotinylated. And then the addition of these primary antibodies followed by the addition of an HRP secondary antibody, which then can help mediate the third step of the workflow, which is an HRP-mediated reaction in which the Allura reagents then becomes a fluorophore that binds covalently around the primary antibody, so around the original location of the target of interest. So as I said, this is a covalently bound fluorophore that will then be resistant to any sort of further stripping steps. So this is a cyclic labeling workflow that then gets repeated for every single target that wants to be detected. It takes around 15 hours if we think about a 9-plex total labeling time. And if you are thinking on doing this with a high number of slides, we do recommend to use automation solutions. So any different kinds of robots that exist, or the automated stainers that exist, for example, the Leica Bond or the Ventana stainer. In any case, the idea here is that you can detect any target irrespective of the target expression levels, which I think is one of the greatest benefits that the Allura amplification workflow has. So you are really able to use any primary antibody, so it gives a lot of antibody flexibility. It results in a very bright signal because you have a lot of fluorophores that are depositing in the vicinity of the target. And it also gives you the opportunity to save a lot on primary antibodies. So we have demonstrated that it can be very successfully used with dilutions that are up to 50K. Of course, there are some drawbacks associated with the workflow. No workflow is perfect. And in this case, it's primarily associated with the labeling time. So because it's a cyclic labeling, so you have to detect one target at a time. So of course it takes longer than if it would be one kind of that all of the targets would be detected at once. And it is important to also note that it is a covalently bound dye. So therefore, if you are thinking on combining this with other type of detection strategies, always it's important to remember that Alluras are done first, because they are covalently bound. They will be resistant to any sort of stripping steps that will come afterwards. And then kind of you can use Alluras with the EVOS S1000. It's not exclusive to the EVOS S1000, but the important thing to remember here is that it does provide a great range of flexibility in terms of targets, not only on the type of targets, but also on the expression levels of the targets. So now I think it's important that you can see what the combination of the Allura special amplification reagents and the EVOS S1000, what that can result into. And here in these videos that are playing, you can see the incredible detail on the tissue microenvironment that you can have access to by using Allura special amplification. The first thing that I want to highlight is that you're looking at two different sections that have been stained with 9 different targets in each case. Of course, because DAPI is present in both cases, then you see altogether 17 different targets. I think you can very well appreciate the brightness of the labelings, the capability to identify very clear single cells, and also the diversity of the targets that we are exploring. They can be low or high expression, and they can be very different in the nature of the target. You have here structural targets, you have many different types of immune cells, so to understand the tissue microenvironment in the sense of the immune environment, the diversity of the immune response here. You can also have proliferation targets, very specific tissue traits that you want to image, and that can be done in a kind of very convincing manner without having to be worried about the expression levels of the target. So this is a very kind of easy way to highlight for you that the spatial amplification reagent solutions that Alluras are bringing provide really a lot of target flexibility to map out the tissue heterogeneity, irrespective of the expression levels of the target. There is a second option in addition to the Allura spatial amplification reagents that we just covered, which I really strongly recommend in the direction of accelerating the spatial proteomics workflow. So we are referring to ISC validated primary conjugates, so ISC proven antibodies that are combined or conjugated with organic dyes to generate a primary antibody conjugate that can be added directly into the tissue and bind, that can be directly bound to the target of interest. The greatest advantage that we can see immediately here is that because we're talking about a very sort of direct interaction with the target of interest, this would allow the possibility of adding the whole primary antibody cocktail in a single step. So there is no need for any additional steps in the workflows. It's also a very straightforward in terms of the time. So it takes about 1 hour to accomplish appropriate labeling. And because it's such a simple workflow, then it is really amenable and quite ideal for high throughput environments. It's also very robust and efficient. And as much as I'm telling all the benefits, I'm also going to be very straightforward with the limitations. We are talking about primary antibody conjugates, therefore it's limited to those that we currently have. And the catalog menu that we at the moment have in our repertoire is around 100 different SKUs, which cover around 28 targets. Those targets are sort of very diverse in the terms of immune targets, proliferation targets, tissue-specific targets. So you can very easily go to a website that I have indicated here. There you can query for a specific target of interest. It will be provided information on the clone, on the fluorophores that we have then available. And with that, it will be sort of relatively straightforward to know what it is possible and how you could prepare a possible antibody cocktail. There is another drawback that is associated with the lower amplification, so it's not possible to compare really with an amplification strategy like the Alluras. But I think the important thing here is that we are talking about something that it's very straightforward. It's a single labeling step, that it's very adequate for medium to high expression targets, and that can be very easily combined with the EVOS S1000, and it can provide really an in-depth level of information, as I'm going to show you in the next case studies that we're going to cover. So, as I promised, I'm happy now to show you some of this data that we have generated with the EVOS S1000 and then in combination with the ISC validated primary conjugates. And remember that we are talking about accelerating and simplifying spatial proteomics research. So I'm hopefully going to motivate you to try these different tools that we have developed, because they allow you to do just that. The power of this fluorescence tool, it really is tremendous. In this specific example, you have healthy human colon to the left and human colon adenocarcinoma samples to the right. A 9-plex panel has been used in both cases. It's the same panel, and it's entirely composed of these ISC validated primary conjugates. And if we focus first on the image to the left, so the healthy human colon, you can see the power of this combination, how these 9 targets, or these actually 8 targets with DAPI, can very clearly show and distinguish the 3 basic layers of the healthy human colon. You see the muscularis externa with the SMA with this light blue market, and then you can see the submucous layer, and then the inner layer that the inner mucous layer that contains the crypts, these very typical crypts responsible for the water and nutrient uptake. If we take a close look into the colon crypts, it's the image that you see, the insert that you see there in the middle. You can see that there's a number of interesting cells that pop up and that help us understand the structure of the tissue. You can see there is interspersed macrophages. There are also a number of other immune cells, and it's very noticeable that you see these Ki67 cells indicating proliferation. So those are cells that are constantly replenishing the intestinal epithelium. Then if we move to the image on the right, then the difference is pretty striking. Now we're talking about a human colon adenocarcinoma tissue, and it's very, very obvious how those infiltrating and irregular structures that are marked by PINE-CK, it's a bit of like in the pink color, they are completely kind of occupying the image. There's actually, we know from the quantitative studies that we have performed that there is also almost a 10% increase, a 10 times increase on the percentage of macrophages in these samples, and we know that they contribute to cancer-related inflammation. We also know that there are more FOXP3 cells and CD4 cells that you can also kind of appreciate from the image. In any way, in any of these cases, you can see the very striking differences between those tissue and how this combination of ISC primary conjugates allow you to truly explore the tumor microenvironment and see the differences between healthy and diseased samples. And if we continue with the same images that we just compared between healthy human colon and human colon adenocarcinoma, there's also all the type of studies that can be done with these special proteomic images that allow you to also quantitatively understand the composition of the tumor. So before we were focusing more on just on the regular completely disturbed structure that is present on the human colon adenocarcinoma samples. But now if we focus on one specific area, it's this tertiary lymphoid structure that is marked in yellow. We have also taken a look at what type of cells are present there, and using phenotyping and performing first segmentation and then phenotyping with Halo software, it is really possible to conclude the kind of specific composition that you have in these TLS structures. You see that there are a lot of cytotoxic T cells, so there is a 62% of cells that are CD8 positive, which are, as you know, are really important for anti-tumor response. We also have helper T cells, around 24% of those, and then also some B cells, around 13% of the cells that we have identified there. So it's not only about understanding the differences in the structure of the tissue when it progresses into a certain disease state, but it's also how that quantitatively can be understood with the data that is generated with the images, and that can be done also in different regions of the tumor. Now we have given the specific example of this TLS, but it can be applied to multiple areas that are relevant for the specific samples that are of interest for your research. Another set of important spatial proteomics imaging data that I wanted to share with you, it's the possibility to combine both ISC-validated primary conjugates with tyramid signal amplification, in this case our Allura reagents, to multiplex on a single tissue sample. Here the example is non-Hodgkin lymphoma human tonsil, and we are comparing disease versus healthy tissue. The labeling process always starts with the multiple rounds of TSA labeling, so using Alluras. This is important because, as we explained, they are covalently bound, so therefore they can be resistant to stripping cycles, and the fact that they are covalently bound then ensures that there is no really cross-reactivity with any additional upcoming primary antibody labeling. Subsequently then, the primary antibody labeling can be mixed and added on a single step, and in this manner then you can combine these two methods for a more versatile imaging. You can think about the opportunities to distinguish very clear phenotypes, like in this example, the very contrasting phenotypes between those different samples, and have more flexibility on the approaches that you use and also on the targets that can be detected. All of which can be done easily with the EVOS S1000 spatial imaging system. The on-mixing can be set up irrespective of the type of fluorophore that is used, so irrespective of the type of labeling method that is used. So giving you full control and full versatility for the imaging generation. So far during this webinar, we've been talking about spatial proteomics and the use of the EVOS S1000 for protein mapping. And this is, of course, the most important application that the EVOS S1000 has been validated for, translational protein imaging. But if we think about other possibilities of other applications that you can also perform with the instrument, then I think we need to show you this example, which demonstrate the validity of using ISC validated primary conjugates, not only for protein imaging, but to combine with RNA detection, in this case using our proprietary bioRNA. So in the example that you see on the screen, we are returning to invasive ductal carcinoma model and comparing with the healthy human breast. And the possibility to have this type of multi-omic approach then reveals a completely whole kind of new wealth of information that can be extremely valuable in understanding, again, the tumor microenvironments. So it's not only that the ISC validated primary conjugates are really easy to use, straightforward, robust, simple, one-step detection. It's also that they can be combined with other labeling methods for other targets, in this case RNA, and this type of combined labeling approach are also applicable and really, really beneficial to do with the EVOS S1000 spatial imaging system. Now, for this final stretch of this webinar, I'm really happy to introduce you to a case study, a collaboration that we have had with the microscopy and cytometry core facility at UMC in Amsterdam, in the Netherlands, with the group of Professor Juan Garcia Vallejo. Juan Garcia is also the director of the core facility, and he has his own research that focuses on glioblastoma. Juan's group was one of the first units that were installed in the world, and he has been collaborating with us for the past year or so. He's been using the EVOS S1000 to study glioblastoma multiforme. So this is the most common malignant primary brain tumor type, and he has a very focused interest in understanding the macrophage heterogeneity in this glioblastoma. He's been mapping the whole diversity or the variety of the cellular status of the macrophages in glioblastoma using single cell as well as spatial proteomics, so the EVOS S1000. And it just so happened that today he is presenting these results that he have been able to gather with the EVOS S1000 in a webinar that will be later today during the day. In addition to this research, we have had the pleasure to collaborate with Thomas Van E. He is a team member within Juan Garcia's group, and we've been working together to develop panels that can be used out of the box, ready to go for whoever is interested in trying to understand the tissue microenvironments. And for this type of panels, we've been using ISC validated conjugates, both these ready-made conjugates that we have discussed in our previous slides, but also conjugates that can be generated using ready labels. Ready labels, it's conjugation kits that we didn't have the opportunity to cover today during the webinar, but I would really strongly recommend that you consult our web pages so you can see in those cases when we don't have the ready-made primary conjugates, then you could utilize your own primary antibody and conjugate it with any of the Alexa Fluor and Alexa Fluor Plus that we have available. So Thomas have been working with us in two panels that you see here on the table, and to be able to shorten the optimization times, we've been using tumor microarrays, and these tumor microarrays have been a fantastic tool for us to be able to screen across multiple panels, to be able to do panel optimization in a very simplified and accelerated manner. And now I'm going to show you some of the examples of the data that has been generated. And as a reminder, all of this work has been done with the ISC validated conjugates, so cocktails that can be added directly into the tissue, and they can be ready to go after 1 hour incubation. Well, the first set of data that I want to show you using these TMAs, it's healthy tonsil, so it's probably the most widely used reference tissue. For us, it's been really important to be able to optimize this panel and go through all the basic optimization conditions using tonsil. You can see here panel number 1, so it's 9-plex, all composed of these ISC primary conjugates. And what I want to highlight, well, what you're looking at is 2 square millimeter images, area scans, and I think that it's pretty striking the fact that signal to noise ratios are really high, and this demonstrates really the applicability and the specificity of the signal that you can obtain with these primary conjugates. This is a very interesting example, lung adenocarcinoma, and I think you can agree that the images here are also pretty striking. We see a very high expression of CD163. It's a marker for M2 macrophages, and also a relatively high expression of CD68 cells, so also macrophages marker. It's quite interesting, and in this specific tissue sample, PD1, it's quite high, and of course it's not surprising that pan-CK has a very high expression. But I again want you to appreciate the fact that this is a cocktail combination that can be added to the tissue in one step. Then even though we are talking about really highly close fluorophores, so very closely overlapping fluorophores, the EVOS S1000 is able to resolve the signal and generate quite specific discrimination between these fluorophores, resulting in these high signal to noise ratio images that are shown here. Last but not least, I want to show you this example of prostate cancer using the same 9-plex panel with this ISC primary conjugates. Again, you can see a fantastic response in CD163 markers and PD1, and we also have a very nice staining with CD8 positive cells. So it offers you the opportunity to map out the entire tissue microenvironment with, of course, within the restrictions of these 9-plex panels, but giving you a very nice overview of the diversity of the population, and also with really striking signal to noise ratios. So I'm coming to the end of our webinar today, or almost to the end of our webinar, and I want to maybe just give you like a summary of what we have discussed and highlight the most important aspects of this EVOS S1000 spatial imaging system. So we've talked about how flexible it is. We are not restricting the use of the instrument with proprietary vendor solutions or with very specific labeling methods. I'll show you very many different examples of how you can use it with pyramid amplification, but also with ISC validated conjugates. It covers a variety of different fluorophores. These labeling methods can be combined. They can also be combined with RNA detection, so leading the way to multi-omics approaches, and the EVOS S1000 really is compatible with all of this. It's also simple. We usually say that spatial biology is not simple, but we have tried to make it simpler, and we have focus on improving how spectral technologies can be utilized. Our mixing workflow has been streamlined, and we have added a level or an additional layer of confidence and transparency with the spectral quality metrics report, so that provides you with a much more clear outcome, and even with the possibility to assess the quality of the data before it's been generated. And all of that, it's making the acquisition in a really fast way. So we show you some of the videos where you could see how fast a 9-plex can be acquired. I roughly can tell you that a single round 9-plex 1 square centimeter scan at 20x can take about 45 minutes, including saving, and the outcome would be a fully unmixed stitch OMET file, and the resolution of that image will be roughly 325 nanometers per pixel, so giving you the chance to delve into the tumor microenvironment. We hope that we have shown you that we are trying to make spatial proteomics easier. and we are kind of giving you tools that cover the entire ecosystem. So we've really tried to design the instrument and the labeling solutions to streamline and simplify your journey into spatial proteomics imaging. And now coming officially to the end of this webinar, I hope that this edition of the Fluorescence Academy was useful to you, and that hopefully you got to learn some of the new tools that we have developed for the spatial proteomics research. If you need any more information, or if you're interested in a demo request for the instrument, please go to our web pages. And if you want to have access to much more comprehensive information on all of our spatial biology resources, please go to our hub, our resource center. You will be able to find there a multiplicity of content. It's been a pleasure for me to be here with you all today, and I want to thank you for joining us, and I'm happy to answer any questions that you might have. Remember that we are here to partner with you, and we will be happy to support you in anything that we can in your spatial proteomics journey.
All right. Thank you so much, Adyary, for your informative presentation. We will now go ahead and move into the Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please do so now. Just type your question in the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's go ahead and get started here. The first question that we have says, can you elaborate on the compatibility of the EVOS S1000 with commonly available labeling methods and dyes in the market?
Yeah, thanks so much for the question. This is one of the questions that we get asked the most. So the EVOS S1000 has been designed in such a way that we can primarily detect any fluorescent labels, irrespective of the amplification, irrespective of the labeling method that has been used. So we can use spatial amplification as well as direct primary conjugates that are kind of labeling the tissue. So we do have, of course, we have highlighted our own solutions, in particular the Allura spatial amplification solutions and our ISC validated primary conjugates. But the general coverage that the EVOS S1000 has, it includes all of the Alexa fluors, all the Alexa fluor plus, our own Allura spatial amplification reagents, which I kind of really recommend if you don't know what the target expression levels are, and as well as opal amplification dyes. So we do have a broad coverage of dyes and labeling solutions for spatial proteomics research.
Okay, fantastic. The next question that we have here says, can you save the 9-plex protocol / dye setup so that it can be used in the next experiment?
Yeah, that's a really good question, and maybe I take this opportunity to highlight a few things of the actual imaging workflow. So our recommendation is that you don't need to know even beforehand as a user if there is a requirement for a mixing. So our software is set up in such a way that the user can simply select the fluorophores, and if a mixing is needed, then it will be automatically toggled. So we also preset the channel configuration for the user so that the user doesn't need to make any decisions. So you as a user don't have to care how the detection is performed. But we do recommend that the first time that mixing a protocol that requires mixing, so that the first time that is set up, that you would acquire this single color control so that we can perform spectral extraction. And once that's done the first time, then the protocol containing this unmixing matrix will be possible to reuse as many times as you wish. Of course, if you change the fluorophore, then we do recommend that you perform a new protocol and repeat the protocol. But for the most part, if you are keeping your fluorophores, then you would be able to reuse that protocol. And if the unmixing metric was generated, then you would also have this quality metrics report that will always give you confidence of the sort of the the possible results of the mixing before the image acquisition has even started. So for us, I think it's very important that the user knows that they can reuse the protocol and they can always have the transparency of the results associated with the mixing metric within the protocol. So the short answer is yes, and then you get a lot of benefits from having the protocol with the mixing metrics and the quality metrics reports.
Perfect. Thank you so much. The next one that we have here says, can the S1000 work with tissue microarray samples?
Yes, it can. So as I think we have kind of alluded that there are multiple ways in which scientists want to accelerate accelerate spatial proteomics workflows, and tumor microarrays are very handy on that respect. So yeah, the system does support tumor microarrays. You need to draw area scans in the individual areas where the tumors microarrays are located, but yeah, it is perfectly compatible with TMS.
All right, wonderful. What are the slide sizes compatible with the EVOS S1000?
So that's really good that there is this question, because we didn't really talk much about it. So the EVOS S1000, it's fully, so it supports, of course, conventional slides. We have a slide holder, a bezel holder that supports 4 slides, so that 4 slides can be placed, and those can be 26 times 76.2 millimeters, so up to that dimension. So we have some flexibility on the dimensions, but you can simultaneously image 4 slides at a time, so they can be placed. We are of course working on the possibility to increase this capacity in the near future, but it is kind of from the perspective of what we currently have, it would support up to these dimensions that I just mentioned. I maybe didn't kind of highlighted this during the webinar, but this can be a good opportunity to highlight this, is that the instrument comes preset with 3 high-resolution size objectives, so 2.5 for overview scan, 10 and 20. So 20 is kind of the most widely used, but you can also, the user can also customize further the instrument, so it can add 5x, 10x, phase or non-phase objectives, as well as 40x. So there is some customization opportunities for the user. The filters do not need to be customized. They will be always to support any of the current as well as future dyes that might be developed in the space.
All right, wonderful. It looks like we have time for one more question here, so we'll go ahead and wrap up with this one. This one says, is the S1000 an upright or an inverted design?
It's inverted design. So most of all of the EVOS instruments are inverted design microscopes. So yeah, it's a member of the EVOS family. So.
All right, fantastic. Well, thank you again, Adyary, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thanks so much, everybody. Goodbye.
Bye.